This is an effort that's been ongoing. I've been taking a lead on this with Mandy Fosang, who couldn't be here today, but we've put this together in association with all the other associate editors. So, as Fred said, we want to improve overall transparency of what's appearing in the journal so that all these issues are not a problem with our papers, to avoid these issues of irreproducibility and avoid this fallout that happens in political circles and public circles and perhaps affects how sympathetic they are to funders. And as Fred, we can skip through this very quickly, there's a lot of reasons why there may be irreproducibility, from method descriptions, poor reagent descriptions, and inappropriate interpretation of the data, and this is what we want to avoid. And I'll just point you at this website at NIH. So there's a big effort at NIH to increase rigor and reproducibility, and a lot of journals are involved in this, including the JBC, and this website has some videos and situations on it for you to look at and get some more exposure to this. And a lot of the issues that I'm dealing with here are dealt with on there as well. So our goal is to improve transparency by improving methodological descriptions, including the reagents, and clearly defining reproducibility and how data is shown, so that it's easy for the reader to assess and for the reviewer to assess during the review exactly what the data looks like, so that nothing's obscured. And so, just to quickly run through, so a lot of what I'm going to describe really is in our instructions to authors, which are available on the website. And if you're going to submit a paper, you should be looking at the instructions to authors for any journal, and looking through what they have to say and what they require you to provide. And in the case of biological materials, we have a pretty explicit list, whether it's DNA sequences, antibodies, cell lines, animals, and transgenic animals. You've got to describe fully what the materials you're using in your experiments are. It's no good telling us what the transgenic mouse was if you don't tell us what the background is. It can have a big effect. The second thing I want to talk about is this issue of experimental uncertainty in statistics, and whenever I think about this, I'm reminded of this phrase that was popularized by Mark Twain many years ago and was attributed to a British Prime Minister, Benjamin Disraeli. There are three kinds of lies: damn lies, and statistics. And I think anybody that's seen statistics in some papers will understand where this came from. And I think that we want to avoid this, obviously, and I think the real key is the transparency in how you report your data and how you have done this statistical analysis. So again, from our instructions to authors, it's critical that you include information on certainty and reproducibility of the data in the figure legends. Don't make a general statement in the methods, because we don't know whether that applies to every figure or not. It needs to be in the figure legends. And again, in the figure legends, you need to state the numbers of independent samples, biological replicates, and technical replicates that are analyzed, and describe how many times each experiment was analyzed and repeated, And then you should very carefully document the variation and precision in your data using measures such as the standard deviation, which is what we greatly prefer, and I'll show you why in a minute, confidence intervals, or the standard error of the mean. So this is why we prefer the standard deviation. So this is from a review written by the guy that developed the GraphPad Prism software suite, published a couple of years ago. And he made up this array of data here, which is shown in this scatter plot, and then just basically analyze this using some commonly used methods. And you can see there's a pretty broad spectrum of the data, as you would expect from many biological experiments. Nothing wrong with this data. There's a broad distribution. And what many people will do is use what's on the right here, the mean and the standard error of the mean, to report that data. And you can see how that does not really faithfully reflect what the experimental data looks like. And so, again, going through these different options, that's why we prefer the standard deviation in graphical data, because this gives a better sense of the breadth of the data that's actually been obtained in the experiments. And perhaps the best method, I think, is the box and whisker method, which gives you a lot more information, gives you a lot more indications of variability. And better still, perhaps, is to use the scatter plot. then you can see all the data. You can still report the mean and standard error on top of it, but if there are other issues related to particularly animal studies using animals, so you want to state whether or not the animals or samples analysed were randomised and indicate how they were randomised before you did the analyses, whether the studies were blinded, that is, did the person doing the experiment know the genotypes of the animals, for example, or the treatment conditions for the animals, and if so, indicate how this was done. And again, sometimes for perfectly legitimate reasons you can have outlier results in your experiment, and so if there are legitimate reasons to exclude data, but if you're doing that, you need to explain how that was determined, how you showed that the exclusion was legitimate, and what the criteria you used were. So again, instructions to authors: we strongly recommend the use of scatter plots for all your data. You can overlay the mean and standard error on top of the individual data points if that's something that's valuable to you, and oftentimes it is. And if you've got a really large data set with a huge number of data points, maybe a scatter plot is a bit overwhelming, and then we think the box and whisker plots are far better at reproducing the totality of the data rather than just relying on a mean and standard error measurement. So this is what I would recommend very strongly, and our reviewers are paying attention to this now. And then showing comparisons between different groups of data. Another issue arises when you start to use bar graphs, plotting the mean plus the standard error of the mean to compare different experimental groups. So again, this is a very nice article written in PLOS Biology last year. And again, this is a data set where they've got what looks like a nice set of experimental data where you've got whatever that is, 20% increase in some measurable outcome with some pretty nice standard error of the mean errors there. And I think if we looked at that data, all of us would want to believe that this is what the data was reporting, where you've got a general increase and a similar distribution. However, you can get this same bar graph from very different patterns of data within this graph. So all of these data sets give you the same graph. And again, what we want to see are scatter plots for both sets of data with the mean plus the standard error of the mean. So you can see, the reader can see, the reviewer can see that this is the data that it really looked like, and it looks like a convincing thing. And if you see things where there's an outlier point or there's some bimodal distribution, that can tell you a lot of information about the biology going on, and it's something the reviewers want to see, and it's something the readers may want to see. And so again, the issue is to be transparent in what you're reporting and report the actual data through the use of scatter plots. So I want to come back now. So another big effort we've had is with Western blots. And Fred dealt with a lot of these issues, and so in our instructions to authors, one of the major issues is the specificity of the antibodies that are being used. And so we call for authors to define the species of origin and source of all antibodies used, including the catalogue numbers and perhaps even the lot numbers that you're using in your experiments. And a lot of people don't keep track of lot numbers, but there are examples where companies will make bad lots where they detect the wrong protein with a lot of antibodies, and so sometimes it may be critical. And if your paper is being reviewed by an expert in the field who's studying that protein, they may know that. So sometimes it's worth keeping. I would encourage you to keep your lot numbers in your experimental notes to avoid any ambiguity down the road if it should come up. If you're using a novel antibody that's been generated just for this paper, this is the first report of this antibody, you need to carefully describe how that antibody was generated, including how the epitope or antigen was prepared and purified. In all cases, whether it's a commercial antibody or a newly generated antibody, you need to describe data supporting the antibody specificity. And if it's a post-translational modification or a neoepitope, you need to document that it is a specific antibody. And obviously the type of information you need to provide is a lot stronger here for a newly generated antibody, but even with a commercial antibody, I'm sure many of you who do Western blots are well familiar with the issue that commercial antibodies don't always work quite like the company says. And so you want to document then why are you convinced that it's detecting the band that the company says it's detecting. And in an ideal world, you demonstrate that some genetic or other molecular modification to the antigen will actually remove the band of interest from your gels. So if it's a post-translational modification, maybe mutate the phosphorylation site or whatever site's modified, or do a siRNA or use knockout tissue to demonstrate that your band, the protein of interest that you're detecting with the antibody is actually disappearing when you remove the gene for that protein. So again, Fred dealt with some of these issues. Immunoblots should be cropped in a way that retains information about antigen size and antibody specificity. Cropped images should include molecular weight markers above and below the band of interest. And you should avoid splicing lanes together. Fred went through this in some detail, and I think what I really want to point out here and emphasize, I can't emphasize enough, is that you keep the original image files, whether it's the original X-ray film on an ECL exposure or the original files you get from your imager apparatus. You save the unadulterated original file and make copies to do any of the manipulations you're changing, so that any of the manipulations you do into contrast or whatever, so that you can always go back to that original source file. Another set of issues arises when you try to do quantitative analysis on Western blots. And so you need to explain how you're doing the quantitative analysis. So how are you measuring the signals that you're seeing on your film or your image? You need to demonstrate or tell us that you've done the appropriate controls to show that the signal intensity is linear with antigen loading. And you need to talk about how the protein loading was normalized in your quantitative methods. And I'll draw your attention here, particularly to ECL type detection methods, that there's a very limited linear range for a lot of these methods. So you need to be very cognizant of that. And this is something I've run across in my own lab, where people are just starting to do Western blots, and they come to me with these things saying there's no difference. And I say, well, you've just overexposed it. You need to get it down into the linear range. And oftentimes I'll get junior people to do their own linear range determination just to make sure that they're doing it correctly. We strongly prefer that signal intensities, if you're looking across a gel at many lanes from a cell extract, trying to compare levels, for example, that the signal intensity is normalized to the total protein loading. And this is best assessed by staining the actual membrane you're blotting for total protein using something like Ponzo S or Amido Black. And we discourage the use of housekeeping proteins, so a lot of people will normalize to actin or GAPDH or tubulin or some other marker protein, and we certainly don't encourage that. And really, if you're going to do that, it's really critical. We want some evidence that the housekeeping protein you've chosen is not altered by your experimental manipulation. So one of the best examples I know of is people that use F-actin for a normalization control, and they're studying something like cell motility, where actin's polymerizing and depolymerizing potentially, and maybe going into a soluble extract or a particulate fraction. And so you could see changes in the levels there for reasons that are that would be making it an inappropriate control to use in this type of experiment. And then if you're using antibodies that are specific for a phosphorylation site or some other post-translational modification, you should normalize those signals to the total protein. So this is, I said, one of my students took the initiative very early in his career. He was not very satisfied. He didn't really believe that the Western blot technique was very linear. He wanted to really validate this, and this is something he did early in his career. So this is just showing Ponzo S staining of total protein on membrane from a brain extract. This is a log scale, and he did 10-fold and 100-fold dilutions, showing the drop in signal intensity on the staining membrane. And again, that's pretty linear across a fairly wide range. So we like Ponzo S. And then this is another example here where he's done a different loading of total protein. This doesn't project very well here, but this is Ponzo S staining. And then this is a Western blot for a ubiquitin ligase. And again, across different volumes of sample loaded, this is a fairly linear relationship here. So this is the sort of result you should be trying to get. And then you're in good shape using your methods for quantitative analysis. So, and this is really important. Instructions to authors. If there's anything that looks funny with the quantification, anything that looks funny with the images, Our reviewers are being trained and to look for these sorts of issues, and if the error bar is too tight, just too tight from an experience point of view, and particularly if representative blots don't really support a tight error measurement, it's quite feasible for reviewers to come back to the associate editor, that's me or Fred, during the editorial review, and they may ask the authors to provide all the high resolution images of the original uncropped immunoblots, unprocessed immunoblots, the details of the quantification, how you establish linearity, and the antibody validation data if there's some issue about the specificity. So again, keep your original data and your unprocessed scans and have that on hand, and make sure you're doing a good job keeping your lab notebooks to avoid this issue, or so you can respond to this issue. So this has got moved around a little bit, but so this is how we're doing this. So right now we're trying to clearly define what we expect and the policies we have as a journal to improve in this area. We're in the middle of educating reviewers and educating authors about these changes in the policy. So on Saturday I gave an expanded version of this talk to the editorial board, which is about 750 people who are our reviewers for almost all of our manuscripts. And so we talked about how these things are going to be implemented by all the reviewers. And we're here today doing the author education part. We hope that you realize that these are just good science. There's nothing special about these requirements. It's just a way of doing good science, and that's what we want to see at the JBC. And we hope that you'll all submit your papers to the JBC and know that you're going to get a good and fair review and it's going to validate that you've done good science in getting it published. And if you're going to do that, I would really encourage you to look at these editorial policies, documents and instructions to authors on the journal website. Fred's alluded to them as well. This is very much a work in progress. I anticipate we'll probably make some more refinements of things and add some additional criteria. And so if you're writing a paper, you want to be sure you're looking at the latest version of what we expect of our authors and what we expect from the papers we're going to accept for publication. So I'd encourage you to do that. And I think we're going to hold questions until after the next speaker. So thank you very much.